Bismillahirrahmanirrahim. The topic of our discussion is hemochromatosis. So hemochromatosis is caused by excessive iron accumulation inside the body. The disease occurs in two forms, which could be either primary, also known as hereditary, or it could be secondary. Hereditary hemochromatosis occurs as a result of congenital anomalies in iron metabolism, whereas the secondary hemochromatosis occurs due to excessive administration of intravenous iron. Total iron in the body in normal conditions is almost 2 to 6 grams, whereas in hemochromatosis, the iron levels can rise to as much as 50 grams, out of which almost 1/3 is stored inside the liver. Apart from the liver, other sites for iron accumulation include heart, synovial joints, pancreas, and skin. One thing you need to remember about iron metabolism is that there is no excretory system for excessive iron in the body. So what happens is the body maintains the iron levels by inhibiting iron absorption from the gut. So what happens in case of hemochromatosis, there is abnormal regulation of iron absorption. Iron is continued to absorb from the gut despite the high concentration of iron in the blood. So this abnormal regulation results in increased iron absorption and increased iron in the body. This excessive iron is deposited inside the liver and certain other organs. There is continued absorption of the iron resulting in almost 0.5 to 1 gram net accumulation of iron per year. So once the total iron in the body reaches above 20 grams, the disease starts to manifest itself. The manifestation of disease could be due to the organ that is affected, such as liver, pancreas, skin, and joints. So how does this iron overload actually develop? Once there is excessive iron absorption and increased iron load inside the blood, this excessive iron is detected by a certain set of proteins. These proteins include hemojuvenile receptor, transferrin receptor 2, and HFE protein present on the cell surface. So let's say this is a hepatocyte, this here is hemojuvenile receptor, this here is transferrin receptor 2, and this is HFE protein on the cell surface. So once there is excessive iron inside the body, it is detected by HFE protein, hemojuvenile receptor, and transferrin 2 receptors. These proteins result in activation of a specific protein known as hepcidin. This hepcidin is released from the cells into the blood and they are transported to gut. So this hepcidin released from the hepatocytes binds to ferroportin channels in the GIT enterocytes. So let's say this here is an enterocyte, this is the luminal surface, and this is basolateral surface. The ferroportin channels are present on the basolateral surface of the enterocytes. This here is a ferroportin channel. So what happens here is that the hepcidin binds the ferroportin channel, and as a result there is internalization of the ferroportin channels. Therefore, there is decreased iron absorption in the blood, thereby maintaining the appropriate iron levels in the blood. So what happens in case of hemochromatosis? These genes, including hemojuvenile receptor gene, transferrin receptor 2 gene, and HFE gene, are mutated. In almost 70 to 90% of the cases, there is a point mutation in HFE gene. The mutation results in replacement of cysteine amino acid by tyrosine amino acid at 282 amino acid site. In certain cases, the gene encoding hepcidin known as HAMP gene, also known as HAMP gene, is mutated. Therefore, a dysfunctional hepcidin is produced, which is ineffective to inhibit iron absorption from the gut. Therefore, there is an increased iron absorption resulting in hemochromatosis. So excessive iron inside the cells damages the cell by lipid peroxidation. This lipid peroxidation is catalyzed by iron inside the cells, and peroxidation means reactive oxygen species attacking the unsaturated lipids inside the cells. These lipids are present inside the cell membrane and certain lipoproteins which are performing vital functions inside the cell. So damage to these lipids and lipoproteins result in hepatocytes injury and ultimately cell death. Moreover, chronic inflammation as a result of excessive iron accumulation also activates the stellate cells. These stellate cells result in increased collagen deposition and fibrosis of the tissues. Moreover, free iron and reactive oxygen species formed as a result of oxidative stress individually attack the DNA, causing damage to the nucleic acids, resulting in mutations. These mutations increase the risk of hepatocellular carcinoma by almost 200 folds. The histological picture of a liver affected by hemochromatosis reveals golden yellow colored hemosiderin deposits inside the hepatocytes. As you can see here, these are the hepatocytes containing a golden yellow colored material which is known as hemosiderin. Hemosiderin is stored form of iron. In addition to hepatocytes, these hemosiderin particles are also present in the Kupffer cells and epithelium of the bile ducts at the later stage. Since iron is inherently damaging to the hepatocytes and other cells, there is inflammation inside the liver. But one specific feature is that there is absence of inflammatory cells at the early time. Once the disease progresses, there is also deposition of fibrous septa within the hepatic tissues, and in the late stages, the disease progresses to liver cirrhosis. The cirrhosis in hemochromatosis is typically micronodular, which means the size of the nodules is less than 3 mm. At the initial stages of the disease, the size of liver is increased, but as the disease progresses to liver cirrhosis, the size of liver is decreased due to shrinking of the liver attributed to fibrosis. Iron is deposited in certain other organs, such as the pancreas contains hemosiderin deposits in the SNR and islet cells. The SNR cells secrete the digestive enzymes, and the islets of Langerhans secrete insulin and glucagon. The damage to the islets of Langerhans result in diabetes mellitus. Moreover, the pancreatic tissue also reveals diffused interstitial fibrosis. Diffuse means present throughout the tissue. The heart is typically enlarged due to deposits of hemosiderin inside the myocardial fibers. These deposits give a brownish color to the myocardial fibers. The fibrosis inside the heart tissues is delicate. Moreover, the dermal macrophages and fibroblasts present in the skin also contain hemosiderin. There is no hemosiderin deposition in the epithelium of skin, but through an unknown mechanism, hemochromatosis results in increased melanin production by the skin epithelium. So melanin inside the skin and hemosiderosis give a slate grayish color to the skin. The discoloration and pigmentation in the skin is mostly in the sun-exposed areas. Hemosiderin deposits in the synovial joints results in acute synovitis, which is clinically determined by joint pain. Moreover, there is also atrophy of the testes. One thing to remember is that there is no deposition of hemosiderin inside the testes, but the atrophy is actually occurring due to a dysfunctional hypothalamus pituitary axis, which results in decreased production of gonadotrophins, and as a result there is atrophy of the testes. Hemochromatosis predominantly affects males rather than the females, the ratio being 5:7:1. The disease has a silent and slow onset, and it becomes significant in 4th to 5th decade of the life. At that particular time, the disease is associated with certain other features apart from the liver disease. The signs and symptoms include hepatomegaly, which is due to the iron deposition and inflammation of the liver, and there could be a history of abdominal pain. Abdominal pain could be either in right hypochondrium or it could be in the epigastrium. The skin shows slate grayish colored pigmentation in specifically those areas which are sun exposed. Due to hemosiderin deposits inside the heart, arrhythmia could be also present along with cardiomyopathy. The most common cause of death in hemochromatosis is due to cardiac disease. The deposits in the pancreas and islets of Langerhans result in diabetes mellitus, and within the synovial joints result in joint pain or arthritis. So due to dysfunctional hypothalamus pituitary axis, there is decreased production of gonadotrophins, which is manifested as in females results in amenorrhea, which means complete absence of the menses, and in the males it is manifested as impotence and testicular atrophy. Due to damage to the DNA by iron and reactive oxygen species, there is a 200-fold increased risk of hepatocellular carcinoma. One thing particular about hepatocellular carcinoma is that the treatment of hemochromatosis does not decrease the risk of hepatocellular carcinoma, since the damage to DNA has already been done. Screening for hemochromatosis could be done by assessing serum ferritin levels, and biopsy can be performed to take a more brief look at the liver and iron deposits in the hepatocytes. The treatment of disease at early stages is with chelating agents. So this brings us to the end of discussion about hemochromatosis. If you have any questions, do let us know in the comment section. Thank you.